In this video, I want to cover how genes and their alleles work together to express all the traits we see in organisms around us. So first, a little bit of review. Remember, DNA is known as the genetic code of life. The DNA in a particular cell is not one long piece, but it's separated into smaller pieces called chromosomes. And depending upon the organism, the number of chromosomes will vary from just a few to, I think there's over 1,000 in fern. Humans, as I've said, have 46 chromosomes divided into 23 pairs. On the DNA, we have nucleotide bases, and the bases are given capital letters, so they're known as A, T, C, or G. And those bases, or the order of those bases, provide the directions for different proteins. And these directions are what we know of as genes. RNA, of course, is what transcribes each gene and then translates it into the correct protein at the ribosome, as we discussed in protein synthesis. So as we've already said, the DNA is organized into chromosomes, and sexual reproduction creates offspring that have chromosomes inherited from both parents. Now remember that sexual reproduction only means that you have two parents to create one offspring. It doesn't necessarily have anything to do with sex, since plants undergo sexual reproduction and fungi undergo sexual reproduction, and single-cell organisms in pond water may be undergoing sexual reproduction. So these pairs of chromosomes are known as homologous pairs, and each copy out of the two in the pair will have genes for the same proteins. Now, proteins go on in an organism to create certain physical traits, and so if we're talking about traits produced by a gene, we're really talking about how the protein then works in the organism to produce some kind of characteristic. Now, the genes that produce this particular trait can be similar or slightly different, and we call these genes for the same trait alleles. So that's what the word alleles means. It's genes that may be slightly different, but they all are determining the same trait. So we have an illustration here on the right of a homologous pair of chromosomes. One came from one parent, one came from the other, and you can see the areas where the genes are been highlighted in different color, and they do different traits, like this one over here. The very first one on the left is the gene for eye color. And we can assume from this picture that parent 1 gave the eye color blue trait, whereas parent 2 gave a brown trait, because they've got some different colors here. And we've got some other traits that are outlined regarding how the body works and temperature regulation over here on the right, or along with some other physical characteristics such as fur texture or how much fur is produced at all. So these are just examples of how, and we've been able to do this with the human genome now. We have been able to map out where the genes are and are beginning to really get a good understanding of what those different genes do and how they're connected to different traits. There's some words you need to know when we're talking about alleles, because you're going to hear me using them. The first word is homozygous, and this means that the two copies of the allele are the same. So they're the same allele. Homo, of course, means same. Then you have heterozygous, and in this case we have two different alleles are present. So same gene, but two different variations on how that gene would be expressed in a trait would be heterozygous. The word genotype refers to the alleles that are found in the genes, and we can't figure this out, without undergoing some pretty sophisticated tests. If you're just looking at an organism, what you're seeing is the phenotype, which are the traits that are shown visibly by this organism. In order to determine the genotype, you can either undergo some genetic running a DNA sample, which is, costing money, or if you have time, you can just produce offspring from that organism and try to figure out what the genes are by what happens with the offspring. Now, alleles, because you have two of them, they interact inside an organism, and they interact in one of several different ways. I'm going to talk just really on 4 different possibilities. There is simple dominance, incomplete dominance, codominance. Do you see a theme here? And then what happens with multiple alleles? Let's start with simple dominance. In simple dominance, you have two alleles, and one is dominant, which means it is going to be expressed as long as one allele is present. And the other one is recessive, which means it will be hidden by that dominant allele unless both alleles are the recessive type. When we write them, when we keep track of what sort of genes are, what the genotype is, what alleles are present, dominant alleles are written with a capital letter and recessive alleles are written with a lowercase letter. You may have done this back in middle school with the Punnett squares and using those different letters. So when the dominant trait is being expressed, the genotype could be homozygous dominant, which means both alleles are dominant allele, or heterozygous. It doesn't matter because the phenotype is going to be the dominant trait. I mean, it doesn't matter so far as how it's expressed. And when it's a recessive trait being expressed, there's only one possibility. It has to be homozygous recessive, because in order for the recessive trait to be expressed, you have to have both copies of it present in the organism. So this is all figured out. The simple dominant system was figured out by a monk, by an Austrian monk who was a scientist as well as being a monk. And he worked in the garden in his monastery, and he studied the inheritance of traits in pea plants. And thankfully he picked pea plants. He had some divine guidance on working with those particular plants because they use simple dominance for the traits that he was looking at. If he picked another organism, he may not have been able to figure things out. But really, Gregor Mendel is the one that's credited with developing the different laws of inheritance on how genes are passed down. Even though in his own time he was not recognized, he really was not given the credit for his investigations until many years after his death. But this system of inheriting things because of simple dominance is called Mendelian genetics. The pea plant traits that he looked at were these 7 traits: seed shape, seed color, pod shape, pod color, the flower location, and the plant size. And in this chart, everything that's here on this top row are all the dominant traits, and everything then on the second row are the recessive traits. So he would do something like this. He would take a pure purple plant and a pure white plant, and when they were crossed, and this is what he was doing before he figured out what was going on. We can see what's going on in the Punnett square, but of course he didn't Punnett's named after another scientist, and Mendel didn't have this to figure out. But he was looking at these plants, these pure purple plants. He'd only ever seen purple plants for generations from these seeds, and so he figured that was the only trait that was present in those plants. And so when those parents, one purple flower was crossed with purple flowering plant crossed with a white flowering plant, and he would do the fertilization by hand to make sure there was no, he had everything very much under control. But the first generation, which is known as the F1 generation, would also all be purple. But if those plants, seeds from those 2 plants were crossed, were allowed to breed, in the F2 generation, in the second generation, there was this white flower showing up again. And that again is showing that recessive trait coming out. As you look at the genotypes listed in this Punnett square, you can see our homozygous dominant, the 2 capital P's, and the heterozygous dominant, 1 capital, 1 small, all producing purple flowers on this pea plant, and then our recessive 2 small P's showing the only one that could produce white flowers. So that was simple dominance, but things are a little more complicated. Another common type of allele interaction is incomplete dominance. Again, you have 2 alleles in incomplete dominance, but neither one dominates the other one totally. In the heterozygous genotype, where you have 1 of each, actually creates a new phenotype that is a blending of the 2. And this is really common in flower color. For example, many pink flowers are coming from a red and a white parent. So instead of getting red flowers or white flowers when you have a parent, one of each, you get a blending of red and white to create pink in the heterozygous genotype. It's also true about fur length in rabbits. If you have a long-haired, a long-furred parent, a long hairs on the rabbit fur, and a rabbit that has got short hairs on its fur, and you cross them together, you'll end up with a medium-furred offspring. You also see this in dog coats and dog tails, that if you have long and short with your two parents, you're going to end up with a medium in the offspring. So this is incomplete dominance. There's a blending in the heterozygous genotype. And then you have codominance, where again you have two alleles and neither one dominates the other, like in incomplete dominance, but the heterozygous genotype, instead of being a blending, a new phenotype, it has both traits showing up. So if it's a flower color, you'll see both colors present in the flower, not blending the two. So we have some camellias down here where red and white are going to give you the red and white seen in the flower. So neither one dominates. It doesn't come out to a pinky color, but you've got sections of red and sections of white. Or we've got a cow over here that has the roan coloring. And of course, this cow had a brown parent and a white parent, and in them they had these two colors in a codominant situation. They are both present in their own individual sections, not blended together, but still showing that color fully, just not over the whole animal. And then finally, there's this multiple alleles or even multiple genes, which are when you have more than two alleles or more than one gene that contribute to the phenotype. And within those multiple alleles, they can have either a simple or a incomplete or a codominant relationship. So it really can get complicated, and that's why until we could do the kind of genetic analysis that we're able to do now, we really have had a hard time figuring out what was going on with some types of genetic inheritance, because it really can be quite complex. But just as an example of this, human blood type has 3 alleles, the A, B, and O allele, and it creates 4 different phenotypes, A, B, AB, or O, because A and B have simple dominance over O, but A and B have codominance over each other. So if you look at this chart, that's of course in very small letters down here, but you can see that a phenotype of blood type A can come from either a genotype of AA or AO. So that A has a dominant relationship over O because this heterozygous, this second one, A and O, giving you an A phenotype, tells you that it's a simple dominance relationship. But the blood type AB is from inheriting both an A gene from one parent and the B or allele and the B allele from the other parent. And so that is a codominant situation. And then our recessive down here of an O, again, fits that simple dominance relationship. Another example in human is human skin color. Skin color is controlled by multiple genes, perhaps several hundred different genes contribute to skin colors, and that's why we have such a variety in skin color present in any population. And then we get to epigenetics, because the expression of genes, the traits that we see, also can be strongly influenced by environmental factors. And this is really where we're seeing some groundbreaking research and some exciting things are happening. So things such as what you eat, your diet, your amount of activity level, the exposure of toxins, both things like pesticides or radiation, and then just sort of general levels of stress and anxiety in your life can all contribute to how your genes are expressed. Epigenetic factors have been found to both turn on genes, good or bad, or turn off genes, good or bad. So if you take this little illustration on the right, if we think this person kind of comes into life and they have 2 choices. They can eat a better diet with good amount of fruits and vegetables, and I would say probably stay active, and so they will remain relatively lean. They're not going to have any side effects on disease, such as what would happen perhaps if they had a fatty liver, and they will pass on good genes, to their offspring. They potentially will not cause any mutations in their own sperm-producing cells, and so it can be passed on to the next generation. Nice, healthy set of genes. But if that same person made some other choices when it came to diet, or perhaps had a job that exposed them to toxic chemicals, then they're going to have a different phenotype being expressed from the same genes, and that might have impact on disease states that are later on in life and could potentially affect their offspring. So epigenetics, I've talked to you about what you choose to eat and how you choose to move, and it really does matter. It can make a big difference in your life and influence things that your genes are not your destiny. You can influence your genes for good or for bad by how you live the rest of your life. So just in summary, the genes in your DNA code for proteins which produce traits. Different variations of the genes are called alleles. Offspring produced by sexual reproduction inherit 2 alleles of most genes. Sometimes they're both the same or sometimes they're different. There's one exclusion to this because genes that are found only on sex chromosomes for organisms that have 2 different sex chromosomes, like humans do with X and Y, there are some genes that are only found then on one chromosome, so you only could inherit one copy. But the alleles you inherit are your genotype. These alleles can interact with each other in several different ways, and I talked about simple dominance, incomplete dominance, and codominance, and that more than one gene or more than one allele may contribute to a trait. The allele interaction results in a particular trait that can be observed in the organism, and so that is our phenotype, and that's pretty much what we're working off of most of the time. And then other environmental or epigenetic factors can influence gene expression so that your genes are not your destiny. So this covers what I wanted to present to you to kind of give you an overview of genetics so we're ready to move on to the next section.